Thanks very much to the organisers for inviting me to speak here today. Just want to check, can everybody hear me?
Yes, maybe you can put it presenting mode.
So let's do this. And can people see my
Yes,
pointer going around the screen there?
yes.
You can, very good. So yeah, I was listening to the talks from Cork yesterday and I rushed to make up this particular slide here because I thought it was important to make the point that the majority of the current phage researchers at university, or sorry, at Cork, that's not only MTU, Teagasc and UCC and the APC, but they all date back to the work of Charles Daly and colleagues back in the late 1970s and early 80s who worked on phages which were affecting starter cultures in cheese. And the majority of current researchers can identify or can indirectly trace their researcher supervisor, I suppose, lineage back to the majority, not everyone, but lineage back to Charlie Daly's group at UCC. So I thought that was important to mention. And Charlie was my own PhD supervisor. He was also the PhD supervisor of Colin Hill, who Liam mentioned in the talk yesterday, and who still, Colin is still doing great work on the intestinal bacteriophages. So that was important to mention, I thought. Now, an overview of what I'm going to speak about today is first, I'm going to just mention a number of the phages that we've considered over the years, some new interesting phage genera that we identified, phages with new features, etc. Then I'll move on to peptidoglycan hydrolases that we've studied, and also delivery of some of these endolysins. I've worked with a number of collaborators at Cork: Olivia McAuliffe in Teagasc, Paul Ross at Teagasc and UCC, Colin Hill at UCC, Jim O'Mahony in MTU, and Horst Neve doing all of my electron microscopy down through the years as well. And I've indicated the various genera of bacteria where bacteriophages have been used in these various collaborations. So I want to acknowledge all of the contribution of these workers here. Because of the breadth of these collaborations, we covered quite a lot of different genera of bacteria, which you see here on this. I won't have time to go into all of them, but I'm just going to mention some of the PhD students and the typical format of the projects that were done. Obviously, the students would have evaluated the various applications, depending on what they were. Obviously, different bacteria present different problems in different environments, so the applications are quite different in each case. All of the students would have done genome characterizations, alignments, etc., and identification of any novel phages, looking for unusual features, new taxonomic groups, etc. The principal students that would have worked on these would be Gary O'Flynn, Sarah O'Flaherty, Sarah Arroyo Marino, Lorraine Anderson, Maureen Henry, Laura O'Connell, Lisa O'Sullivan, Marianne Horgan, Aidan Casey and Vincenzo Pannone, and Colin Bottomer. I'm just going to pick one of these, Colin Bottomer, to give an example of some of the great work that was done. And in addition to that, allows me to acknowledge some of the collaborations that established throughout Colin's project. What Colin did was he isolated a number of soft rot enterobacteria which were responsible for diseases like blackleg and soft rot in the potato crop, economically quite important in Ireland. And he also isolated soil samples from potato harvesting machinery and used these soil samples to successfully isolate a lot of phages which targeted these Pectobacterium atrosepticum pathogens of the potato plant. These aren't all of Colin's papers, but they're ones which show an emphasis in finding new types of phages. You can see a number of genera have been identified. Colin also published some other papers as well. So showing you these papers gives me an opportunity to acknowledge the collaboration of Rob Levine's group. You can see he's a co-author on a lot of these papers here. And it also allows me to acknowledge the contribution of this great bioinformatician, Alan Lucid, who has contributed considerable work to our phage program as well. He died tragically in 2017. I'd also like to acknowledge the Oxford conference here, because Colin presented his work on this particular topic here back in 2017, and that allowed us to develop a collaboration with Dan Turner, who happened to be here that day, and also, linked Colin up with the various taxonomists like Kropinski, Adriansons, etc., and allowed us to come up with these papers and define where these different bacteriophages that Colin identified would fit in, and allowed us to propose a number of new genera, which you see here defined on the International Committee of Taxonomy of Viruses and these documentations here. 3 of Colin's phages, CB1, CB3, and CB5, targeting Pectobacterium atrosepticum, were defined as a new genus within the family Scytoviridae. Another one, CB5, became identified as a new genus, Phymonovirus, within a new subfamily, which we were allowed to name ourselves, and obviously we named it after the location where the phages came from, and these became Cork Virinae, this new subfamily. And you can see that since then, I just downloaded this yesterday. It shows you the large number of members, genera, that are included in this particular subfamily. Another aspect of Colin's work was looking at Acetobacter baumannii phage, which also identified in a new genus of metrivirus. So that's some of the work that we did in terms of new phages. Colin also identified a jumbo phage, which was reasonably new at the time, with a lot of hair-like particles on the contractile tail of the myovirus, which was also interesting as well at the time. When you're using whole phages to eliminate undesirable bacteria, one problem that you'll encounter again and again, and you see it throughout the scientific literature as well, is that bacteriophages tend to have a very narrow host range. There are, of course, some exceptions, some broad host range phages. And one phage that we had a long time ago was phage K from Staphylococcus aureus. People considered that a broad host range at the time, but with all of the collecting of hospital isolates that we did, we were able to find lots of strains which were resistant to phage K. So it's quite a challenge finding phages which will cover an entire genus. Resistance obviously would be due to things like the absence of adsorption molecules on the surface of the bacteria, or the presence of various phage resistant systems like adsorption inhibition, injection blocking, restriction modification, CRISPR-Cas systems, and abortive infection. And this is a huge area in itself, characterizing these various phage-resistant systems. I just point one thing out: when restriction modification, these systems tend to be very, very common in Staphylococcus aureus. It's of course possible to overcome these by passaging phages through them, and phages become modified and then become capable of eliminating the RM containing bacteria at higher efficiency. And a student of ours reported how this would be done in expanding the range of coverage by phage K by making a number of restriction modified variants of it. Another thing that you can do, which we did, was you can broaden your mixture of phages by looking for additional isolates. I'm just going to hop to the next slide briefly. This is phage K plaqued on a number of Staphylococcus aureus strains, different MLS multilocus sequence types that have been isolated in hospitals all over Ireland. So these are all the MLSTs that exist in Ireland in the various MRSA infections. And when you look at phage K, you can see it's not very good against a lot of them. So what we do then is we take these particular resistant strains, and just in a collaborative project with Nina Chanishvili at the Bacteriophage Institute, we started fishing out phages from principally the Fersisi mix from Tbilisi, and we were able to find phages that filled in all of the gaps in this table here. So having 4 additional phages from these combined with phage K, then we were able to cover everything. Obviously, it's interesting to look at the genomes of these new phages, and we found that they were all very, very closely related to phage K itself. This isn't all of them, but it's a typical result that you get with these two phages here, B1 and JA1, showing you how closely related they are to phage K. And this is an ongoing topic. We've been cloning the various open reading frames that differ between one genome and another to try to elucidate the differences between the host ranges of these. That's work in progress. We haven't identified anything yet, but we have been cloning these in trans to see what their effect is on phage replication. So that's the issue of host range. Everybody who's using phages as biocontrol agents encounters this. Another issue that can arise is, depending on the ultimate destination of your phages, there can be things in the environment that might inhibit your phages, prevent them from eliminating your target pathogen. And one good example that we found here is in a raw milk environment. Staphylococcus aureus phage K doesn't work very well against its homologous host. In other words, the bacterium that was used to propagate the phage, when you do the challenge in raw milk, the interaction doesn't work well at all. And it's problematic due to a kind of a clumping effect that happens to the bacteria due to the presence of immunoglobulins in the milk. So this is problematic. And you can get similar things in other environments as well, various things that negatively impact on the interaction between bacteria and bacteriophages. I would make the point actually that a lot of research papers will report on showing that bacteriophages work in milk. I have never seen a case where they've done this with milk that has not been heat treated first. The heat treatment always eliminates this negative effect of the immunoglobulins, and it looks like it works fine. It never works fine, and it's totally irrelevant to say that bacteriophage kill bacteria in milk when you've only used a heat-treated milk to demonstrate it. So that's one shortcoming of a lot of papers that mention these kind of things. When we identified that a number of years back, that is what really led us to using peptidic glycan hydrolase technology. It circumvented some of these difficulties, particularly the host range difficulties. Peptidic glycan tends to be constant right across a particular genus, and therefore differences in receptors just don't matter at all. The target of the endolysin will be the same right across the genus. The endolysin, as you know, is the enzyme which is expressed at the end of the lytic cycle. There's also a peptidic glycan hydrolase, which facilitates the entry of the genome of the bacteriophage into the cell as well. So there's a number of very common enzymatic domains that have been identified across the various endolysins reported in the literature. And this is the endolysin from phage K up here. It's got a cysteine, histidine-dependent amidohydrolase peptidase, an amidase, and this classic cell binding domain as well, which is very important to anchor the phage endolysins in the peptidic glycan area to make sure they work where they're meant to work in the natural lytic cycle. You don't want the endolysins leaking out, killing other potential target bacteria in the natural process. This is just a generalized peptidic glycan cell wall here and shows you where these enzymatic domains operate. This is a phage tail hydrolase showing again a CHAP domain and a lysozyme domain as well. So we've worked with a number of these endolysins. And when we started off first, we were using Qiagen vector systems for purifying them and histag and cation exchange chromatography to purify them in our own lab. We moved on to using PET vectors in E. coli, which were a bit more efficient. And more laterally, we've started to not do it at all ourselves. We would simply send the nucleotide sequence of the endolysin to this company in the Netherlands, and they would manufacture the endolysin for us and send it back. And they would also send back, obviously they do QC analysis on it as well, send us back the data, and also sent us back the E. coli clone in case we wanted to make any more ourselves. So that's a very, very good system, and I highly recommend it, particularly because of the QC that's done. It can save an awful lot of time in your own lab. Looking at activity of the endolysins, the routine method that we use is polyacrylamide gel electrophoresis, where the acrylamide is impregnated with autoclaved bacterial cells, which give it an opaque texture and allow you to see where the endolysin is active. You can also see your endolysin activity by adding some endolysin to turbid cultures of your target bacterium. This is Staphylococcus aureus here at a titer of 10 to the power of 9 CFU per mil, and the endolysin has just been added there to the tube on the left. And you can see that within a relatively short space of time, it actually takes 5 minutes, this video is going to speed it up, you see complete elimination of the culture in the tube on the left. So endolysin, yeah, they're very, very easy to work with, quite well defined, not like bacteriophages, which are, you know, every bacteriophage is different in its own way. So endolysins are far, far nicer systems to work with. All endolysins, depending on where they're going to be applied, you'll need to know their pH range, their temperature range, and their tolerance to various environmental parameters. You'll also need to know its, you know, shelf life, how sensitive it is to freeze thaw, and good ways of long-term storage of these enzymes. So we've typically done that with our endolysins, and these are just profiles of results with these CHAP endolysin from bacteriophage K from Staphylococcus. In vivo assays, this is one good example here of a Staphylococcus aureus bacteriophage endolysin being used to eliminate Staphylococcus aureus from the nasal system in animals. Nasal carriage is a very, very important source of Staphylococcus aureus bacteremia in hospital infections. So we used a bioluminescent Staphylococcus aureus strain. You can see it here on this using the in vivo imaging system on an agar plate. And you can also see it after colonizing animals with it. We've colonized the nasal cavity here. So we set up this experiment where we took 14 animals colonized with the bioluminescent Staphylococcus aureus. 7 of them were untreated, they were the control group, and 7 were treated with the CHAP-K enzyme for 1 hour. And you see with the treated animals, you get complete elimination of the bioluminescent Staphylococcus aureus strain, whereas it's still present in the control group. And 100% removal of the Staphylococcus aureus from these animals was verified by traditional bacteriological methodologies, which I don't need to go into. We also made sprays of it and used it to eliminate bacteria, Staphylococcus aureus, from skin as well, and typically showed a 2 log cycle reduction in carriage of Staphylococcus aureus on skin. This is pig skin here, and this is again using the in vivo imaging system, but obviously we also verify our data using traditional bacteriological techniques, which is the best way to do it really. In terms of the host range, as I've mentioned already, the target of endolysins, generally the peptidoglycan, is conserved throughout the genus. So look at these Staphylococcus aureus here. These are Staphylococcus aureus from the MLST groups from hospitals. These are EPS producing Staphylococcus aureus, and these are staphylococci of species other than the species aureus. You see they're all other non-staph aureus staphylococci, and the bars here represent delta OD. So this is effectively showing the extent of reduction in optical density. So the CHAP-K endolysin is very, very effective, pretty much equal throughout all of the genus Staphylococcus aureus, and also working a little bit on close relatives on the phylogenetic tree where there are some similarities in peptidoglycan structure. I'd also like to acknowledge the help of Jackie Cooney with some computer programs that she had in University of Limerick, which enabled us to develop an in silico model of the CHAP-K endolysin. We were very fortuitous with this particular endolysin in that we were able to see that there was the blue color indicates a positive charge. There was a strong positive charge on this endolysin. It's not something that generally happens with endolysins, but this one was particularly strong. And that's very advantageous because staphylococcal cell walls tend to have a negative charge. So there's a good attraction between enzyme and substrate, in addition, obviously, to the active site, which was fortuitous. This work was done by PhD student Mark Fenton, a student of mine 10 years ago. I'd also like to acknowledge a collaboration with Mark Van Raj at the CSIC in Madrid, who took this work a stage further in elucidating the X-ray crystal structure of this endolysin. And this was excellent. It was done by a joint PhD student, Marta Sanz Gaitero, who, through the X-ray crystallography, was able to define a number of regions that were unresolved by the in silico analysis. So that was a great collaboration, and that's published in a number of papers there some years back. What I want to talk about next is some of the biofilm work, some of the biofilm targets that we used for the endolysins. One thing that's very special about biofilms, obviously, as we all know, they're a source of various bacterial infections in humans. They're quite problematic because bacteria in the biofilms tend to be more resistant to antimicrobial agents. There's a physical sort of protection against antimicrobial agents, but also bacteria in biofilms, they're not in the log phase of growth. They're generally closer to the stationary phase. They're quite sessile in these environments. And there's considerable physiological changes that occur in bacteria, which contributes to their higher level of resistance against antimicrobial agents, not only phages, phage endolysins, and also antibiotics, of course. We made a number of modifications to ChapK, generally site-directed mutagenesis, which allowed us to learn a lot about the enzyme, what residues were important for various, in the active site, or for holding various metal molecules in place, etc. This is one modification that we made with the ChapK endolysin, which enhanced its activity. We took the cell binding domain from the molecule lysostatin and fused it to the ChapK endolysin from phage K, giving us this chimeric protein down here. This was done by a student of mine, Sara Arroyo Marino, and the chimeric protein showed enhanced activity by comparison with the original ChapK endolysin. This is just log phase cells, and you can see there's a more rapid reduction in optical density when the chimeric protein is used. With 7-day-old cultures, if you look at these two concentrations here, which are closer to the MIC levels, you can see with the chimeric protein shown here on the right, you're getting some activity at 5 micrograms per mil compared to ChapK, where there's little or no activity, and with the 20 micrograms per mil, a much stronger activity than you see with ChapK. Also, you tended to need more of ChapK to see equivalent amounts. In terms of biofilms, biofilm prevention assays, ChapK also was not as good as the chimeric protein. The biofilm intensity is shown obviously by the blue stains, which show surviving biofilms treated with various concentrations of either enzyme. The chimeric protein is more effective at preventing biofilms. Whether it's log phase or using a 7-day-old culture to start to generate your biofilm, there is a difference, but the chimeric protein is more effective. This one here is biofilm disruption assays, and the green bars here are ones which use the 7-day-old cultures to generate the biofilms. And again, with the chimeric protein, the optical density of the biofilm after the treatment is much, much less with the chimeric protein than it is with the ChapK itself. Obviously, the difference also is seen with when you use the log phase cells to generate the biofilm. That brings me to the final stage in my talk, which is looking at the delivery systems for endolysins, getting them to their appropriate area where you want them to work. And there's 2 different modes of delivery that we've been looking at. Using secretion vectors, i.e. delivery from bacterial hosts, or also using nanoparticle technology. First of all, looking at secretion technology, we've used this vector PNZ8048. It's a lactic acid bacterium vector. This is the essential structure of it here. It's got a promoter and a signal peptide from Lactobacillus brevis, and downstream from that we've cloned the endolysin from the ChapK enzymatic domain from phage K, and we transformed this construct into Lactococcus lactis, and we've plated it out on an agar plate here, which has been seeded with Staphylococcus aureus. And you can see the result of the secretion of the endolysin here on the streak on the right. This is Lactococcus lactis secreting ChapK, whereas the one on the left, control, you don't see any zone of inhibition. So in principle, at least, the system works. So let me briefly look at where we use nanoparticle technology for endolysin technology. For this endolysin, I'm going to come back to secretion systems again in a minute with other endolysins. This is a collaboration that we did with Toby Jenkins at Bath University, and Toby was able to package ChapK into polyisopropyl acrylamide nanoparticles. And with this particular molecule, you can modulate the temperature at which the nanoparticle will disrupt and release its payload of endolysin. So combining the polyisopropyl acrylamide with allylamine allows you to strictly control the release of endolysin from the nanoparticle. These are control cultures here, 3 controlled cultures with no chap in the nanoparticles, and you see the three replicates. There's no release of endolysin, obviously. Where the nanoparticles contain the endolysin at 32 degrees centigrade, you're putting, this is actually a dressing with nanoparticles fused to the hospital dressing. They drop the dressing into a turbid culture of these bacteria. At 32 degrees centigrade, the bacteria survive. At 37 degrees centigrade, the endolysin payload is released from the nanoparticles, and you've complete elimination of the bacteria. And again, traditional bacteriology was used to verify the difference. And there's a 4 log cycle reduction in the cell number of bacteria when the culture is shifted up to 37 degrees centigrade. So I want to come back to nanoparticles, come back to secretion systems again here. In the context of Clostridium difficile. This is a bacterium which is naturally antibiotic resistant. It's present at low levels in a lot of colon, in the colon of lots of people. When people are taken to hospital and given antibiotic therapy for various infections, the antibiotic therapy will eliminate a lot of the competition that C. diff would encounter in the large intestine. You get the bacterium rising to higher numbers and giving rise in many cases to pseudomembranous colitis, which is a very, very serious condition. So
Adrian, you have a few minutes left.
I'll rush my last few slides. Not much.
Okay, wonderful.
So basically, yeah, this is a C. diff phage isolated by a PhD student, Marianne Horgan. She cloned the endolysin, demonstrated activity anaerobically by measuring counts. You see, it's an amidase endolysin, as you saw on the last slide. Basically, yeah, we put this into a delivery system as well, initially PQE60 and then later into PNZ. The ultimate aim being to get it into a probiotic, getting the probiotic to release it in the intestine. Obviously, you're not limited to lactic acid bacteria. There is a probiotic E. coli, famous E. coli Nissl, which is also a more convenient system to use. We did get successful secretion, but we consistently found, as we did with the ChapK earlier, secretion level is too low to be anyway meaningful. So at this point, we became aware of work of Danish Malik, who was using a pH-triggered nanoparticle system and for releasing phages. And this nanoparticle system, which is based on alginate, it's modulated such that the nanoparticle disrupts at a pH 7. So if you look at the system we're developing here, the nanoparticles are fed to the intestine, which is a low pH, and then once it gets to the colon, which is a pH 7, the pH rises, the nanoparticles release their payload, and obviously you're delivering your C. diff endolysin. This is work in progress at the moment, and it's being conducted by Kerry Richards, and there's a paper being prepared at the moment. So that's going well. Another area, it's a collaboration with a company called CC Bio, and Sarah Arroyo Marino cloned an endolysin for Gardnerella vaginalis. This is a uropathogen, which is tending to become quite antibiotic resistant. And the work we've done with this endolysin, indeed a family of these endolysins with CC Bio, has led to a publication which just came out a couple of months ago in collaboration with Ronan McCarthy at Brunel University and also CC Bio, a spin-out company. So that's gone quite well. And the last 2 endolysins Lactobacillus causes spoilage in yeast propagation tanks in breweries. A Lactobacillus enterocolic is an ideal way of keeping the numbers down. And another one here, Propionibacterium enterocolic, amplified from a prophage of Propionibacterium, a phage of Propionibacterium. And yeah, we did identify 2 endolyses. In the latter 2, they turned out to be insoluble. That's work in progress. We need to go back to the drawing board again with that. So that concludes my talk, essentially. I'd like to acknowledge the various people that I mentioned throughout my talk, and also funding agencies, Science Foundation Ireland, the Department of Agriculture, the various fellowships that I got from Teagasc down through the years, and my various collaborators at UCC and Teagasc as well. And of course, I'm a member of the Elementary Thermobiotic Centre at UCC, so I'd like to acknowledge their support down through the years as well. So if you have any questions, I'd be glad to try to answer them.
Thank you. So do we have a few questions? Something in the chat, maybe?
Hi, my name is Emma Farquharson. I work at Folium Science. This was a fascinating and well-paced a presentation. I was wondering, for when you encapsulate these endolysins within your nanoparticles, do you find that they would aggregate and that you'd end up, even if you started with a certain concentration, your loss then came from this aggregation of forced encapsulation?
That's a very good question. I'm not a nanoparticle engineer, and it's a question for the likes of Danish Malik and Toby Jenkins. These are the people who make them and who would encounter these problems. So it has been reported to me, but really our work is really at its infancy in the case of the pH induced release. So I can't answer your question satisfactorily.
That's all right. Thank you so much.
There's a question online from Janet Nail.
Yes, thank you so much for that wonderful, amazing job and work you've been doing at UCC. My question is on the testing of the endolysin and the ChapK. I was wondering whether you tried a polymicrobial environment or they're all pure cultures, especially with C. diff. We know that issues with dysbiosis normally causes infection to trigger in C. diff infection. So just wondering whether you've tried, to see whether these endolysins or the ChapK, interferes with the microbiomes or the specific bacteria.
The quick answer is we haven't done polymicrobial cultures. With Staphylococci, you're generally speaking talking about the skin and the nose. It's quite a specific environment. Obviously, polymicrobial challenges are far more relevant when you're talking about gut bacteria, where there's quite a different dynamic going on. But no, we haven't done it in the case of ChapK.
Okay, and I think with that, we would like to thank you, Adrian, for being with us from Cork.